So hello everybody. My name is Ying Liu. You can call me Ying. A little brief introduction about myself. So I got my MD in China. Then I went to the States, got my PhD. Then I came back to China to join BGI, which is BGI means so contribute 1% to the International Human Genome Project, and that's also how the BGI got started, and how the genome field in China got started. And at that time, our chief scientist, Dr. Yang, is two founders for BGI. One of the two founders of BGI. The other one is Wang Jian, which is our president. And so they two start realize at that time, 1998, genomic field is going to be it's a future direction for human research, for animal plant research. So they start to get money, like our president Wang Jian, get money from his own company and get a loan from the bank and support this project. That's why by sequencers and by other agents, and finally we make it work. And China started to get famous in the world for the genomic field. And later on in 2005, we contributed 10% for the haplotype map project of the human genome. So we started from only a participant to getting more involved in this kind of international projects, which I think every Chinese should be very proud of this. And finally, in 2008, we sequenced the first Asian human genome all by ourselves. Actually, this is from our chief scientist. It's his genome, and we found out have the tendency to get a diabetes in the future. So if you're interested, you can sequence yourself, sequence your own genome, and talk to me or talk to Shenzhen. We can give you a discount for that. And so this is the first Asian human genome, and also all the sequencing part, bioinformatics part, is done by BGI itself. And it's published in Nature as a cover story. We also compare this genome with African genome. Here's the interesting part. So we found about 5 megabase of genome normal sequence, which is not available in any of the reference genome, human genome available. So this 5 megabase of normal genome, they are not junk. They contain some coding sequence. They contain some regulatory sequence. So which means they contribute to the gene expression, contribute to the phenotype. So we predict there are still about 40 megabase of novel sequence in the human genome. So our suggestion is, if possible, if condition allowed, you just do the de novo sequencing, not the resequencing. I mean, for each individual, for each like a human, you do the de novo sequencing. Otherwise, you probably miss some very important information. And so here we find out some of the novel sequence have different tendency to either decline or increase following the migration of the population, African population. So if you're interested in Iraqi people population, you can also do the same thing, like do the de novo sequencing and find out do the population genetics, find out maybe the migration of the population, and also construct the database for that. And so this is just to give you a general idea about human research progress in BGI, all the way from 2000 to 2005, 2008, and finally now we are part of the 1,000 Genome Project, International Human 1,000 Genome Project. And so what's next? After all this, what we are going to do next? Why we are doing this, right? So, so we want to extend it to everyone, which is like personal genome. Which with personal genome, you can have your own personal genome data, and you can do the prediction of health diseases, and you can maybe prevent the disease from happening, and also find out the treatment for the disease. And also, if you want to find out what kind of talent your children has, you may have their children sequenced and find out that may be good at like music or like football, or anything you're interested in, and talk to us or talk to Shenshen, and we'll be very like to help. Okay, so it gave you a general idea about the human research. So from the basic, you do all the genomics, transcriptomics, epigenomics, metagenomics, proteomics. And next, you can construct you can sequence the whole population and construct the database and set up the baseline for the population. For example, right now, we collaborate with some European countries and also some countries from Asia Pacific, and also in local China. We sequenced the human about like thousands or hundreds of human of the population, individuals from that population. And we'll, based on that data, we'll construct the database, and this database will be the reference for the later either research, either clinical service, either personal genome. So anything starts from this database. And many people many scientists, especially top scientists in the country, are so interested in these projects. And BGI initiated this called the Million Human Genome Project. We want to, because we have the resources, so we can construct a database for the scientists, for the population, and we can, develop later research or clinical tests. So anything you're interested. Then we can study the human disease research and discover the relationship between phenotype and the genotype. And also finally, the target is a personal genome and personalized medicine. For example, pharmacogenomics. If you know the genetic background of this human, you can decide what kind of medicine is useful for him or her, right? And if not, you just skip this medicine and find out something else. No waste of time and money, especially if the country have a public health system. It's very important for the government to do this. So that's the future, which is already going on. And one example about how to, for the future use for human resequencing is this all-in-one array. It's designed by BGI itself. We call it super capture array. We collaborated with Nivogen to make this really happen. So with this array, so first you do the sequencing for the population, right? You construct the database. You know all the genetic information for the healthy people, and you can design this chip. This chip can include all different informations. One is the exome region, because if you do this kind of a screening test for the whole population, so it has to be very cost effective, not too much too high price, but very efficient. So you probably it's not so practical to use the whole genome sequencing for everyone, right? So with this chip, you can first choose the exome region only, right? And combine with population-specific tax name. That's where the genetic variation came from this population. For example, it can be Chinese, it can be from Europe, it can be from England, Japan, or Iraqi. And also, you can do customized regions, like HLA type tags in there. And so these are all advantages. You can capture the most valuable 6% of the genome and completely take the disease-related mutations and also unknown mutations. Application, so you can either screen this whole population and contribute to the health system, and or you can help your drug R&D process more efficient, and you can do the population research for different human population. Okay, so a little bit more about the disease research in BGI. So everybody knows different disease they have different backgrounds, right? Some parts contributed by the environments, some are contributed by the genetic backgrounds, the genome. But for each disease, these two factors can have different contributions. Some are more affected by the genetic background, some are more affected by the environment. Here's just a list of the diseases. So one big project in BGI is metabolic disorder research called Sino-Danish project. It's a collaboration between BGI and Denmark. So we are screening patients based on 3 phenotypes: the visceral obesity, type 2 diabetes, and hypertension, and also 2 stages. First, we use exome sequencing to sequence about 1,000 cases and 1,000 controls. Stage 2, you will do the genotyping, select about 20K SNPs, and sequenced in 17,000 samples. And right now, it's still going on and going on very well. And some of the data already published in Nature. So here we identify a lot of genetic variations, either in the exome, in the intron, synonymous, non-synonymous. So we found out actually many of the genetic variations affecting human health are in low frequency. They're not high frequency, in low frequency, which means you probably miss them with the traditional regular genotyping process. You have to do the deep sequencing with NGS to find out these genetic variations. So another example is this spinocerebellar ataxia. This is a monogenic metabolic disorder. So we sequenced only 4 patients from a 4-generation Chinese family, do the exome sequencing and the linkage analysis. So we found out a genetic mutation in TGM6. And also by linkage analysis, we confirmed this mutation is really contribute to the disease. So if you have very nice family for some kind of disease, talk to us, and we will do the analysis for you, and I'll talk to Shanshan. He'll talk more later. And another case is a still ongoing project about autoimmune disease. So the reason I shown this the case is because you can see here, the strategy is different. It's combination of all different sequencing methods: deep sequencing, epigenomics, and transcriptome, iron level, and small RNA. So right now, not only the people are not only interested in sequencing only one level, either DNA or all RNA or protein. They want to sequence at all different levels. And also it's much easier for their publication, and also much more information for their to find out what's really going on there. We call it transomics or multi-omics. So that's a kind of new concept for the genomic field. And we also have a lot of collaboration with the pharma companies. One with GSK. So here we do target region sequencing for 200 genes, and with about 15,000 of people, and sequence about 30x to discover the rare SNPs. Remember I said the SNPs affecting human health are mainly in low frequency. So right now we found out 50,000 SNPs, and some of them are unique and non-synonymous. The other part is ICGC, we call it International Cancer Genome Consortium. So this is international organization. So all different countries, many scientists from all different countries are contribute to this organization. China, which means like BGI, is in charge of the gastric cancer. And for example, also like pancreatic cancer, breast cancer in Australia. So, so here you want to find out the genomic information, transcriptomic information, and epigenomic information. For each, we'll sequence about 50 different tumor types and the subtypes, and 500 tumors per type. So totally it will be 25,000 cancer genomes. And that's what BGI is doing. And we collaborated with Beijing Cancer Hospital and also Peking University. We're collecting samples, do the sequence and analysis, and also maybe some complementary studies. And finally, the data storage analysis and management. It's ongoing. And also, NGS can provide new solutions for drug R&D. Like I said, like we can do, some of them I already mentioned in previous slides. Here I'm not going to too much details, but for example, FFPE samples. FFPE samples are sometimes very precious, and also the DNA is hard to get from them, and usually in very small amount. And so we found out we can do sequencing for FFPE samples, and also we try to minimize the DNA requirements for this technology. And also, for others, like all-in-one array, already mentioned. Oh, the other thing is we can do de novo sequencing for cell line or even single cell. So we just published 2 papers in Cell, 2 sister papers in one journal, one issue. So you can sequence one cell for their genome and find out a lot of useful information. If you're interested, you can ask us, or you can check online by yourself to find out what's going on in these papers. This just very interesting thing is about RNA. So for NGS can use for diagnosis, prediction, or prognosis, or even treatment of the disease. And here, just example for molecular prognosis. So for this disease, we sequenced about 30 patients with longer survival, and the other 30 as a control for shorter survival. And we do small RNA sequencing, and we found out 4 microRNA. These 4, they are different in patients with longer survival and shorter survival, which indicates maybe later on you can, if you want to do the prognosis of some cancer patients, you can do the sequencing and find out their microarray profiling and do a more accurate prediction. This is the EpiTwin project I just mentioned. It's the largest ever epigenomics project in the world. It's a collaboration. It's a three-party collaboration, BGI, King College in London, and Pfizer. So King's College provided the samples, BGI provided the sequencing, the technology, and Pfizer provided the money. So this worked very well. And so we are about to sequence 5,000 twins with MADIP sequencing, not the whole genome by sulfate sequence is too expensive. And also find out the methylated regions, differentially methylated genes correlated with the different disease. For example, adiposity, diabetes, and some other cardiovascular disease and bone diseases. So part 2, I know many scientists today here is doctors, but because BGI is such a big complex, we focused on everything. So I'll talk a little bit more about animal and plant. Hopefully you're interested. Otherwise, I'll go very fast. So here, non-human related. So like I said, genome of one species is always the start of the research of that species. So you can see from here, we sequence the rise and weight, and ever since it's been, sorry, it's all Arabic numbers. I cannot. Anyway, so this is the year we got the genome published for rice, and from then on, you can see the publication on these two plants that go all the way up. So this happens to other genomes too. That's why we are so interested, because like for especially nowadays, for 21st century or later on, genomic information is so important for the country, and for plants, for animals, for everything. So give you 2 demo cases. One is cucumber, the species called Chinese long, and also one is panda. The reason, why we sequence them, because cucumber tastes very good, right? And panda looks cute. So that's the reason we sequence them. And for cucumber, you can see, this is just to give you some general introduction, information about these 2 genomes. And so for cucumber, it's the first genome we use next generation sequencing, first case. But by that time, we are still not so confident for the next gene sequencing. So we confirmed our data with Sanger sequencing. And for the Panda, it's all by Illumina, next generation sequencing. And the time is different. This one takes about 18 months, and this one takes only 8 months. And the cost per base is also different. You can see the cost for the sequencing is go way down a lot. And here's some details. So this is the first case of the novel sampling by Solexa, and we can do the mapping for very important genes and QTLs. And with all these genes and QTLs, you can do molecular breeding later for the plants or animals. And this is a Panda. So actually, the sample is from Jin. It's our mascot for Beijing Olympic Games in 2008. The reason we sequence it is because it's so cute, but actually we want to find out some specific things about the Panda. For example, why it cannot only eat bamboo but not meat. We actually found out it has the meat, gene for meat digestion, but why it cannot eat meat? Because by sequencing, we found out this gene called T1F1 is got deactivated by insertion. This in exon 3 and exon 3 and exon 6. And also, do you know what's the closest relative for Panda by our analysis? It's very hard to imagine. It's dog. So dog is the closest relative to Panda based on the evolution analysis. And we also find out the Panda is actually have a very high heterozygosity rate. So it's not easy for this species to disappear on our planet. And we also sequenced the gene for cuteness. You know what we found out? Nothing. So next, one follow-up project about Panda is when we want to find out, because it does not have the gene for plant digestion, but it can digest bamboo. Why? It must be related to the bacterial flora in its gut. So we did a lot of work on that, but unfortunately so far still not too much progress because the gut have a lot of bamboo fibers, and but bamboo, the genome is still not sequenced yet, and it's very complex, like a 4 gigabase, and only 1% of the sequence we got is from the Panda, from the bacteria, which is not so useful. So BGI now sequenced a lot of different species, and with them, we can actually make a dinner for you. If you come to BGI for a visit, I will show you around and give you a very nice treat. For example, the cucumber for sure, and tofu, very healthy, and cabbage. I'm not sure, people here like meat, right? So now we have chicken and duck, and also some seafood, oyster, and for the main food, like rice, maize, and also the wheat, and some fruits, like a watermelon, sweet melon. And after dinner, we can show you the wildlife world, our panda, and the penguin, polar bear, and antelope. It's from Tibetan. So we call this project the three-pole antelope. So you sequence these animals from all 3 different poles. South Pole, North Pole, and the Tibetan plateau. And also the monkey, and some others, other plants. And we also have our own factory. So by sequencing the silkworm, we know which gene is contributed to the production of the silk. And we know by sequencing the soybean, we may increase the oil production, increase the quality, and also the cotton, and some biofuel plants for the future industry. So based on all of this, we initiated these projects, I think 2 years ago, 2010, to called Library of Digital Life. Another million projects, animal plants projects. We are about to sequence millions of them. Right now, so here it shows 1,000, but actually now we mention like millions, which includes both the de novo and also the resequencing. So we do 2,000, already sequenced 254 species of plants.